Thanks for coming tonight, everyone. Welcome to Breaking Science. Tonight's topic is revolutionary cancer therapies, taught by none other than myself, Jim Sampson, PhD. And you'll notice that the letters in front of my name are not MD. So it should go without saying, but anything that I say in tonight's talk, don't take as medical advice. If you have questions about cancer, about your personal treatment or something like that, talk to your doctor. Okay. So welcome, everyone. Thanks for coming. If you have any questions at any point during the talk, feel free to unmute yourself. You can also type them into the chat line, but because of the way my computer is configured, sometimes I don't notice these things. So feel free to yell at me. So I also want to point out that I do work for a pharmaceutical company. I make cancer drugs. I'm not going to mention any of my work. I'm not going to advocate for any particular company's drug. Nobody knows that I'm here giving this talk. Nobody's paying me, and all of the opinions that I express are entirely my own. So completely independent here. So the outline of tonight's talk, I'm going to start by telling you a little bit about what cancer is. And if we want to talk about cutting edge therapeutics for cancer, we have to figure out how it has historically been treated. We'll have to, I apologize, dive a little bit into the immune response to cancer, as most of the therapies we're going to talk about modify the immune response. And then finally, we'll get into the meat and potatoes of the really cool, brand new therapies that have emerged within the past decade. So what is cancer? And I apologize, there's going to be a little audience participation in the beginning of this talk. So what tissues can be affected by cancer? Oh, this is super awkward by Zoom. Anyone?
Oh, I did not catch on that I was supposed to talk.
Congratulations, Christian. You've been volunteered. What tissues can be cancerous?
All of them.
Yes. Literally anything can become cancer. How does that happen?
The DNA. Oh, sorry.
She knows. She knows more than I do.
Yeah, there's some sort of mutation for a variety of reasons. And is cancer just one thing? Let's see. No, it's not one thing. Good. Even within the same type of cancer, if two different people get breast cancer, for instance, it doesn't necessarily mean that's exactly the same thing. And within the same patient, not all cancer cells are exactly the same. That's really important to keep in mind, as it's going to shape our understanding of how we should be treating these things. Is cancer contagious? This is a trick question. Well, the obvious answer is no. Generally, they're not contagious. The exception to this would be if you were genetically identical to somebody and you happen to bite their face, like Tasmanian tigers do. Probably doesn't happen too much with humans. So the safe answer is no, it's not super contagious. I often get the question, if I make it to some arbitrary age like 70 or something without getting cancer, am I home free?
Nope.
Yeah, of course not. The older you get, the more likely that you will develop full-fledged cancer. And do we have a cure for cancer sitting in some basement somewhere and the government is suppressing it? Please answer this.
5.
No, the answer is of course no. If it were that simple, I'd be rich, and we'd all be rich. Okay, so cancer is the unrestricted growth of cells. Normally in your body, many of your cells divide and replace dying cells. And this is a tightly controlled process. So in the event of cancer, something has gone wrong and these cells divide out of control. Cancer cells sort of lose the specialization that they would have otherwise had. So lung cells are really specialized to, pick up oxygen from the air, and lung cancer cells sort of de-differentiate. They lose that ability to do these really specialized things. And that's part of why cancer causes problems. Cancer also sets up its own microenvironment. So it essentially brings its own food and drink to the party. It brings in blood vessels for solid tumors that deliver nourishment, and it suppresses the immune cells that are present. Either inherited or acquired mutations can cause cancer, and generally one mutation isn't enough to get oncogenesis, the generation of cancer. Usually you need an accumulation of multiple mutations. Now, some mutations are more likely to predict the development of cancer than others. Many of you are probably familiar with BRCA2. This is a gene that's normally a tumor suppressor. It's involved in repairing DNA and killing cells that something has gone wrong in. It has a very high penetrance. So if you have this mutation, a mutation in this gene, then you have a relatively high likelihood of developing breast cancer. It doesn't mean that you're guaranteed to, but it means that you should be monitored and perhaps take earlier action than you would have otherwise. Cancer is actually incredibly ancient. This shouldn't be too surprising, but the more we dig into it, the earlier we find examples. So this is actually an ancient, essentially proto-turtle from, I believe, 240 million years ago, where you can see that this growth right here looks exactly like a periosteal osteosarcoma, so a bone cancer that would be found in humans nowadays. It's actually kind of shocking how similar it looks to cancer that we can find in people. Cancer has been a known problem to humanity for a very long time. The earliest written examples of cancer being diagnosed that I'm aware of are from this papyrus that was originally from probably about 2500 BC in ancient Egypt. Although I wouldn't be too surprised if the cultures in ancient China had discovered it, thousands of years before. So cancer has been a scourge on humanity for as long as we've been human. Cancer is unfortunately very prevalent in humans. Every year, just in the United States alone, there are almost 2 million new cases of cancer, and 600,000 people die of cancer every year, according to the National Cancer Institute. The most common cancers in the United States are breast, lung, prostate, and colorectal. Thankfully, these are not the most lethal cancers. The most deadly cancers are glioblastoma, pancreatic cancer, mesothelioma. These are cancers that we really don't notice until they're further along in development. So generally, the earlier you can detect a cancer, the better likelihood you have of responding successfully to treatment. And like glioblastoma is in the brain, pancreatic cancer is, of course, your pancreas. Mesothelioma is a cancer of the linings of like your thorax and stuff. So we don't have great screening methods for these things. As that technology advances, it's likely that we can decrease the mortality rate by treating these tumors earlier. But cancer is actually the second leading cause of death in the United States, behind heart disease. And you can see that both in men and women as they age, the incidence of cancer goes up really significantly. So are there any questions so far? Check the chat window.
Just
that last chart. Oh, I'm sorry.
No, go.
Oh, okay. That last chart where there's a very noticeable difference between men and women.
Yes.
Is that mostly attributed to like there's a difference between prostate and breast cancer? Or?
Yeah, I don't actually know that. I think that yes, like prostate cancer does contribute to a lot, and obviously it's a high mortality cancer. It could also have something to do with typical lifestyles. I'm not entirely sure. That would be a good question for an MD, to be honest. Any other questions?
Yeah, what I was going to ask is any kind of thing that is an oma is considered a tumor, but not all tumors are cancerous, correct?
Oma is generally the term for a cancerous mass. Not all tumors are malignant.
Okay.
Yeah.
So
Sometimes you can have a couple cells that start expanding and then they just sort of stop based on where they are, or tissue cues. Like there can be multiple reasons. They don't have a lot of room to grow, and those don't go on to develop like full-fledged cancer that will kill you.
Like a lymphoma.
Correct. Well, yeah. So you can have, I mean, if you have a lymphoma, that's cancer, generally malignant.
No, lymphoma. A fatty tissue tumor?
Oh, lipoma. Yes. Yeah. So something like that. That would be generally, to my understanding, a benign growth.
Okay, I'll shut up now.
No worries. Any other questions? All right. So how have we historically treated cancer? This is a fun story. So the first people to think about treating cancer were, not surprisingly, the ancient Greeks. Their philosophy was to not operate on cancer. This was for a number of different reasons. One, they believed the body was sort of perfect as is and shouldn't be cut into. And also, there were, early doctors like Galen that thought that cancer was an accumulation of this liquid called black bile. And so there would be essentially nothing you could do about it. Probably not so much the case. So fast forward about 1,500 years, you have essentially the first guy to try cutting into bodies and really start studying, what's going on inside, William Harvey. Once he started doing autopsies, we could take cross sections of tumors, look at what's growing in there, and start to think about what we would do to treat it. And then fast forward, another 100 years or so, and you have another British guy, John Hunter, who said that this is a great criterion. Tumors can be cut out as long as they're movable. So imagine going to your doctor, they try shaking the strange growth on your side, and if it can move, then they'll perform surgery. Not the greatest criteria you could imagine for determining if your cancer is operable. The other problem, of course, is that surgery in the mid-1700s was incredibly gruesome. There was no anesthesia. Patients would often die of infections. It was not really a great approach. Fast forward in another 100 years, and just down the street, or I suppose this is a virtual talk, so maybe not down the street from you, but at Mass General Hospital, the first use of ether as an anesthetic was done in the mid-1800s. This made surgical procedures a lot more tolerable, and so surgeons were willing to take on more complicated tasks. Soon after, you had this guy, William Halsted. He had the philosophy that if you just kept cutting more and more tissue out, you could cure the cancer. So he pioneered these radical surgeries, radical meaning going down to the root. He tried really hard to cure breast cancer by doing radical mastectomies. Eventually, he would remove so much tissue that the patients would essentially die from from losing important tissue. And he eventually, begrudgingly, by the end of his life, admitted that you could not cure cancer, not all cancers, by just cutting out more and more tissue. But it was a start. Moving on to World War I, a lot of countries used chemical agents, such as mustard gas. We realized quickly that this is a really terrible thing to do, causes a lot of really, really, really terrible effects. And so internationally, we came to an agreement that chemical agents are not really the best things to keep around. We shouldn't be using these in war anymore. But of course, countries wanted to keep it in their back pocket. So the American government and other governments started looking into using mustard gas and other chemical weapons as essentially chemotherapeutics, because they made the observation that they would stop cells from growing in a dish. A great way to make sure you can keep the chemical weapons on hand just in case you need them. Fast forward to London. You have this really nice X-ray machine. And as you can imagine, although X-rays, we still use radiation for tumors, this is a very early example of a machine which is quite open. And a lot of doctors and nurses themselves developed cancer as a result of exposure to the radiation they received. And then shortly afterwards, Sidney Farber of Dana-Farber fame was the first person to essentially undergo a rational drug design clinical trial, where he had these antifolates, which he saw again inhibited cells growth in a dish, and he used them for acute lymphoblastic leukemia. treated a bunch of kids in his very tiny basement room that they gave him at the hospital, and saw some pretty impressive results for the time. So if you'd like to know more about the history of cancer, I've dramatically oversimplified it. I'm going to highly recommend this book, The Emperor of All Maladies. It takes what could otherwise have been a really, really dry subject and makes it really pretty interesting. So any questions on the history stuff? I'm not necessarily likely to know a lot of those answers. Let's see, we got a chat question. Okay, good, yeah. Other people think it's an awesome book. Yeah, it's really well written, very accessible. Antifolates, so they inhibit a growth factor, essentially, that is necessary for the unrestricted growth of the cells. Cool. All right, moving on. Cancer in the immune system, my favorite part.
Okay.
So, the scary thing about cancer is that you have some oncogenic events happening pretty frequently in your body, more frequently than you develop cancer. So normal tissues can be mutated because of carcinogens, radiation, what have you. And then you'll have a couple cells that will divide uncontrollably or unrestrictedly. The lucky thing is generally these cells, once they start dividing like that, express different proteins and signals that will indicate that something has gone wrong. These we refer to as danger signals and tumor-associated antigens. The awesome thing is that your immune system can recognize those and through multiple different mechanisms can lead to elimination of these cancer cells. And for a lot of, essentially precancerous events that occur in our bodies regularly over the course of our lives, this is sufficient to clear the initial development of cancer. So I won't go into all of the details. I would love to, if anyone has any questions about this, I'm happy to talk ad nauseum about any of this, but it's not all super relevant. But basically, your immune system has many, many different ways of killing cells, tumor cells. You have T cells, which you've probably heard of, perhaps even in the context of certain viral infections that are present currently. You also have these things, natural killer cells, which are really, really good at killing, altered self cells like tumor cells. So there are multiple different mechanisms that your immune system uses and that we'll eventually take advantage of for the therapeutics that we'll talk about in the second half. We have a question. Cells at work. Yes, cells at work. There's an anime on Netflix, which is surprisingly accurate. It's an educational anime teaching you about the immune system. I can't believe that I'm recommending it, but yeah, it's pretty good if you want a good primer on basic immunology.
They did have a cancer episode.
Oh, cool. I've only seen the first 2 or 3 episodes just to make sure it was accurate before I recommended it. But yeah, check that out, or you can ask me any questions you want, what have you. So there, like I said, there are a number of different mechanisms that we have for killing tumors. The problem becomes the immune system recognizes those very specific bumps that are different on your tumor cells that are not present on your normal cells. What happens once we kill all of those cells? Well, sometimes the tumors can escape and still be cancer cells, but lose those different things, those things that make the immune system activated. There's a point at which essentially the cancer cells exist at an equilibrium with the immune system. The immune system kills off any really different, really new cells, like this one on the left, but it's sort of okay with these ones right here. It doesn't really know what to do. It can't entirely tell if they're different from you or not. So at this point, your cancerous cells are not growing outrageously. You probably don't have a full-grown tumor that you're really noticing. But this is the point right before escape, where the tumor just grows out of control, expresses all these immunosuppressive mechanisms, and really does everything it can to promote its own survival and develop at this point clinical cancer. So in addition to just losing the things that make them different, as I mentioned, there are active mechanisms that the tumor can express to shut down your immune response. And we'll talk about some of them because these are like directly relevant. So you have soluble proteins that the tumor cells express that will shut them down. You also have like cell contact molecules. So when your immune cells come into contact with the tumor, they say, nah, I'm going to go to sleep. And it's actually a surprisingly accurate metaphor. I would also like to impress on everyone that the role of the immune system in cancer is actually really complicated. It's not just that your immune system shows up and it kills the tumor. Chronic inflammation, such as you would find in the case of like IBD or like a chronic gut inflammation or something like that, can cause the production of growth factors that promote cancer. So IBD, some forms of IBD can be associated with some forms of gastric cancer. And some immune cells that are present in the tumor microenvironment shut off the good or anti-tumor immune cells. So not all immune cells are created equal. Inflammation also helps a tumor to escape the site that it's growing in initially and metastasize. One of the things that the immune system normally does is it remodels your tissue after it's injured. It expresses enzymes that essentially cut up the damaged sections and allow tissue to be patched up. Cancer takes advantage of this and lets the immune system essentially cut it out of its initial site and, flow through the blood vessels and go to wherever its metastatic site is going to be. And some inflammation is also necessary to help tumors engraft at whatever site they're going to. So it's important to understand really the details, not just the question of, is there immunology present at this tumor? So any questions about that? No questions because I've lost everyone, or no questions because it was all super clear?
I have a question about your last slide.
Yes.
So do these factors play into making certain types of cancer harder to diagnose, harder to treat, both?
Yes, certainly harder to treat. So generally, most of the therapeutics that I'm going to talk about in the next couple slides work better on tumors that already have T cells present in them. They're just being shut off by the tumor itself. There are also tumors that exclude T cells. Like it's actually really not beautiful, well, kind of beautiful from a scientific point of view. If you look at these things on a slide or something, you can see that the T cells all hang around on the outside of the tumor growth and don't really go in and damage the tumor. There's also some tumors where there's very little T cell infiltration at all. They're just not present. Those tumors have the most immunosuppressive mechanisms and really don't respond all that well to the next generation therapeutics that I'm going to talk about. But we're working on that. That's actually, I won't talk about the specifics of what I'm doing so I don't sound biased, but that's what I'm trying to do. Okay, cutting edge cancer therapeutics. They fall into three major classes-ish. There are permutations that blur the lines between these a little bit, but there's a class of molecules called checkpoint inhibitors. There's autologous cell infusion, and there are vaccines. So the checkpoint inhibitor molecules, these things work by removing the brakes from immune cells. So it's important to keep in mind that activation of your immune system is actually really dangerous for you. If you have a viral infection or any other infection, you want your immune response to kill the virus or what have you. but it can also cause a lot of damage to your tissues if it continues longer than is necessary. So whenever an immune cell becomes activated, it also turns on compensatory negative signaling mechanisms to shut itself off. So when, for instance, a T cell becomes activated, it turns on molecules called CTLA4 and PD-1. Both of these, if they bind to molecules on the tumor or other immune cells, will cause the T cell to be turned off. Generally, in the course of a normal immune response, these negative signals aren't really enough to shut everything down, but they're a way. There's many different mechanisms of positive and negative signaling that add up, and hopefully, if you still have a virus, the positive signals win. If you don't, the negative signals win. In the context of cancer, cancer is really good at expressing the proteins that bind to these negative molecules and shut off the immune cells, specifically through CTLA4 and PD-1. So checkpoint inhibitors are things that get in the way of this interaction right here. They stop the negative signaling and allow you to only let the positive signaling happen and turn on your T cells, your anti-tumor cells. There are a number of different FDA-approved checkpoint inhibitors. They first were FDA-approved just a little under 10 years ago, so they're all quite new. So there are antibodies that block CTLA4, PD-1, and then the other side of the ligand for PD-1, PD-L1. These already have been approved for at least 11 different indications, 11 different types of tumor, although there are literally hundreds of clinical trials going on currently trying to expand the scope, trying to reach additional patients with different types of cancers using these same molecules. Additionally, people are using them together because in some cases, blocking one pathway is not enough. You want to really, block both in order to make sure you're getting your maximum immune activation. So this is really great, right? This is a halfway decent list of cancers that, otherwise would add up to a lot of deaths. And we've delivered some promising therapeutics for them. How well do they actually work? They work great, but only when they work. So the problem is only about 15 to 25% of patients will respond to checkpoint inhibitor therapy. But for the ones that do, they can have extremely durable responses, that last for a long period of time, which you don't really get so much with chemotherapy and radiotherapy. So one very famous example of a patient that got checkpoint inhibitors is former President Jimmy Carter. He went to his doctor with stage 4 melanoma. He received a course of 4 months of radiation and this PD-1 blocking therapy, and he's essentially been cancer-free in the years since he got the therapy. Problem with that is this is, of course, a very famous case. A lot of patients went to their doctor saying, I want what he had. And, as I mentioned, most patients don't have such a wildly successful response as he happened to. Now, the really important question is how do we figure out which patients will respond, to which therapies? And this is literally, a billion-dollar question. The great thing is we have a lot of different clinical trials going on targeting not only these pathways, but also other checkpoint pathways. But the thing that cancer patients don't necessarily have is a lot of time, right? Like they can't try one drug and fail on that, and then try the next 6 and fail on those until they find something that works. So it would be ideal if we could identify, in advance what patients will respond to what drug. Someone asked, is there data on how often checkpoints are prescribed? I'm sure there is. I'm not familiar with that, to be honest. There are criteria that have to be met in order to get checkpoint therapy. And so for some of these, like the PD-1 blocking antibodies, some of them are becoming standard of care for some cancers. So if it's standard of care, then it's prescribed very frequently. But for a lot of the other indications and for some of the for some of the therapeutics, it's more exploratory. Does the MAB thing? Yes. So MAB at the end of a drug name means antibody, monoclonal antibody. That's a good question. And actually, if we go back to the previous slide, you can actually figure out more than just if it's an antibody based on the name, like atolizumab. I don't actually know much about this, but I can tell because it's Zumab that it's a humanized antibody. It was raised in some other non-human species and then genetically engineered to be a human protein. Yeah, so naming conventions you can pick out a lot of interesting details from. Good question. Yeah, so it would be really great if we could identify which patients will respond to which drugs. So there's a lot of effort both by universities and by companies trying to identify predictive biomarkers, something you could measure in the blood before treatment that would predict success with a particular therapeutic. Unfortunately, there hasn't been a lot of great success so far, but still there's a lot of effort being invested into this, so hopefully something good comes of it. There are a lot of problems with checkpoint inhibitor therapy. As I mentioned, we're removing the brakes from the immune system, and we're not just doing it in the tumor microenvironment. So patients that receive checkpoint inhibitor therapy often have immune-related adverse effects. So these things look like autoimmune disease. They can appear essentially anywhere in the body. And we really don't have a great way of predicting who's going to get what immune-related adverse effects. In general, these can be managed with corticosteroids. It's not ideal because then you get some degree of immunosuppression, but somehow you can suppress with corticosteroids just outside of the tumor and still have enough anti-tumor efficacy for the drug to work. When we combine different checkpoints, toxicities become much more common. So CTLA-4 is the most toxic checkpoint that we have currently. 20 to 30% of patients get really severe toxicities. PD-1 has a less common rate, a less frequent rate of that. If you put the two together, the frequency of grade 3 or 4 tox goes up quite a lot. It's not necessarily dose limiting, like treatment limiting at grade 3, but if you have a grade 4 tox, then you have to discontinue the therapy, which is really unfortunate. So there's also a lot of engineering that's going on currently to try to target these specifically to the microenvironment of the tumor, or engineer the antibody in ways that will reduce the toxicity while still maintaining the anti-tumor efficacy. Additionally, there are tons and tons and tons of additional targets being pursued currently, and literally hundreds of, probably more than thousands of clinical trials ongoing currently, pursuing new targets, things that are not CTLA-4 PD-1, and also combinations with CTLA-4 or PD-1, or trying them in different indications, combining them with chemo or radiation. There's a lot of work going on, and I suspect that all of it will be beneficial or most of it will be beneficial for somebody. Like I said, it's just about identifying which patients will respond best to a particular therapeutic. This is a terrible example. I hate IL-2 receptor. KIT. I don't know, maybe patients with non-small cell lung carcinoma will respond better if they have a particular biomarker. It will take identifying biomarkers that can predict success.
I just want to say something.
Sure.
Jimmy, that I work in pharmaceuticals as well.
Sure.
End user, patient, stuff like that. I get a lot of patients who are like, well, how do I know that this is going to be the best for me? Or things like that. And that's just one of the tough things sometimes that you have to explain to them. It's uniquely your body.
Yeah, there's a lot of effort being put into big data, to really be able to, it would be great if we could take, a blood sample from someone, run RNA sequencing, DNA sequencing, and be able to determine what the best therapeutic for them would be right out of the gate. We're having a really hard time doing that. I mean, it's a very complicated problem, right? Because as you point out, every person's cancer is different. And as I mentioned earlier, even the cancer within one person is not a homogeneous thing. So it's not a trivial problem. I do hope that with enough heads and enough effort, many of these drugs will find a home for different patients. But yeah, that's the biggest hurdle, right? Figuring out who to treat with what. But we are working on it. Just to underscore how impactful these drugs have been, the Nobel Prize in Physiology or Medicine in 2018 was given to Jim Allison and Tasuku Honjo. Side fact about Jim Allison, he's one of the least charismatic speakers I've ever heard, as you may be able to guess from his picture. Not super interesting. But this work, all of the work involving PD-1 and CTLA-4 has essentially been done between the late 90s and now. So this is an incredibly short period of time from initial discovery to Nobel Prize award. So it should really hammer home just how impactful these checkpoint inhibitors have been in delivering options for patients that didn't have any at that point. Any other questions with checkpoints? All right. Autologous cell infusion. So this is a fancy word for making you a GMO. I'm joking. I like this picture because it makes people uncomfortable. Autologous cell infusion is essentially a personalized medicine approach where, yeah, you do kind of become the GMO. What happens in this procedure is T cells are withdrawn from you, and in vitro, so in a lab, you insert a gene for a special type of receptor. This is not necessarily a receptor that's found on any actual cells, but this receptor is able to bind tumor cells and activate the T cell really robustly in the absence of really anything else. So this is essentially putting your immune system on overdrive, right? So once the gene is inserted, you expand the cells up a lot in vitro, so you have a ton of them, and then reinfuse them back into the same patient. So theoretically, the CAR cells, as they're called, chimeric antigen receptor cells, will find and attack the cancer cell, and then you'll be cured, right? So these things work. Normally, a T cell needs to encounter an antigen-presenting cell that has picked up and digested and spat out part of the tumor and can say, hey, look, this is a tumor. You should recognize it and kill it. Additional soluble factors are necessary to activate the T cell and tell it exactly what to do. Then you get an activated T cell. It leaves your lymph nodes. Lymph nodes are present all over your body. They're things like the things in your neck, which feel inflamed when you get sick. It leaves the lymph node and then goes and attacks the tumor. Chimeric antigen receptor T cells, CAR T cells, essentially cut out the middleman, right? They put in a special receptor that can bind the tumor antigens directly. You don't need that antigen presenting cell to let you know that the tumor is there. And it has all of the signaling domains downstream necessary to induce activation of the T cell just when it binds to its tumor antigen target without those additional soluble mediators. CAR T cells have evolved quite a lot since I've been aware of them. They started with very simple cytoplasmic domains, signaling domains that were essentially just what nature has produced, and then they got more sophisticated, putting in additional co-stimulatory signaling molecules. CAR T cells can also be engineered to do additional things, but the first generation of CAR T cells that went into patients was just really putting in this receptor. So these are really brand new. there have been, I believe, 2 FDA-approved cell therapies already, both of which are for juvenile B-cell acute lymphoblastic leukemia. So this is a patient population where really they failed on chemo. They have no other option. Prior to this therapy, this was a death sentence. And these are kids, right? So they really needed something. So the leukemia cells in this case express CD19. It's a protein. So the chimeric antigen receptor T cells, the CAR T cells in this case, target that tumor protein, CD19. Initial results from this type of clinical trial were really, really remarkable. 83% of patients had a short-term, went into remission in the short term. I believe that was at about a year. And then still 43% of patients survived at 18 months. And again, this is a cancer where essentially 100% of the kids would be dead by that time point. There are also in development additional CAR T cells targeting other tumor-associated antigens. They're in trial now. Some of them look promising, some of them look less promising. People are also engineering more sophisticated CARs that can target 2 different things. One of the problems with this type of therapeutic is, just like with a normal immune response, what happens once the tumor loses this antigen? That's probably part of the reason why we don't have a higher overall survival, because the tumor can lose that one antigen and then grow out again. If you could design a CAR that recognized 2 different proteins, it's like giving 2 different antibiotics at the same time. You're much more likely to kill everything and then perhaps get a true cure. People are also armoring. People that make CAR T cells love their cute names. They're armoring CAR T cells, making them more resistant to the suppressive mechanisms of the tumor and more resistant to being exhausted, which is exactly what it sounds like. If a T cell is activated for too long, it kind of just gets tired and doesn't really do much anymore. Kind of like me, to be honest. But like myself, people are trying to armor CARs to prevent that. There are a number of limitations to CAR T cell therapy. Unless you specifically design them to, they really don't go anywhere that normal T cells don't go. And this, as someone asked earlier, are there different types of tumors in terms of what immune cells can get in and such? Yeah, solid tumors don't really do too well with CAR T. CAR T cells don't really treat solid tumors super well because T cells don't penetrate them really well. And as I mentioned, if the tumor can lose the antigen, the protein that the CAR T cell is targeting, the tumor is now invisible and your fancy engineered CAR cell doesn't really do anything. Exhaustion is a problem. Long-term, over the course of years and years, the data don't exist yet because these things are so new, but I believe at this point there are 2.5 year time points for some of these patients, and the success rate has dropped off. It still works in some patients, but it doesn't give you the long-term success that you'd want. And toxicity is an enormous problem because you've essentially enabled the T cell to react anytime it happens to see whatever protein that you've engineered it to react to. So you better hope that that is really, really specifically expressed on the tumor. In the case of CD19, this B-cell lymphoma, CD19 is expressed on all of your B cells. So you do get off-target effect. It's really on-target, but off-tumor effect. And cytokine release syndrome is really, really common in patients that receive CAR T cells. 77% of patients get it. So this is really not a great toxicity. The other big problem with CAR T cells is they're outrageously expensive. And this is essentially by design. I mean, it requires a lot of lab handling. So you need a technician to take out the cells from the body. You need people to be able to do the genetic engineering, culture the cells for a couple weeks, and then reinfuse the cells back into the body. There's really not a great way to do that cheaply. And of course, you need a hospital that's specialized to handle this thing, like NYU or something. Small community hospitals are unlikely to have the appropriate licensing to do this. So there are plenty of people that are trying to make off-the-shelf CAR products, and that would get around the exorbitantly prohibitive cost. You know, you'd still probably need specialized hospitals, but it would get rid of one of the problems. And I'm going to highly recommend, I highly recommend XKCD in general, of course, but they have some really funny cartoons about this, pointing out that you're essentially treating cancer by giving your immune cells HIV. It's obviously not HIV, but a vector derived from HIV is what's used to transduce the T cells. So, any questions about CAR cells? We have something here. Roughly how long does it take to engineer CAR cells that recognize a new target? So the engineering is not incredibly challenging depending on what the target is. If it's something, so let me go back to this. The part of the CAR that recognizes the antigen is essentially a single chain FV. This is like an antibody fragment. So if you already have a single chain FV that recognizes the target that you want, then it's not too hard to engineer them together. If you're going after some incredibly weird marker, like maybe you picked out something unique that's expressed on your tumor in, you know, an RNA-seq or something like that, that people haven't really targeted with antibodies before, then you'd have to launch a campaign to generate a single chain FV, and that would take much longer. Although still, hypothetically, you could imagine getting a halfway decent single chain FV in 6-8 months, putting it all together, doing preclinical testing. That's going to take a while. It depends on how much technology you have access to. If your company or whatever already has, you know, a construct for the transmembrane and signaling domains, and you just swap in the single chain, then it's going to be faster than it would otherwise be. So that was not a super specific answer, but I think the answer depends on who you are. If you're well positioned, then you could hypothetically imagine within 2 years going from you know, a proof of concept to like maybe an early clinical trial. But that assumes that you're well positioned to do that. Let's see. You're welcome. And I trivialize, you know, the engineering. It's not super easy, but for me, people who know how to do it, you know, would be able to do it much more easily than I do. All right, another class of therapeutics, anti-cancer vaccines. You probably don't think about it, but we already have 2 essentially anti-cancer vaccines. These are both vaccines that are against viruses that can cause cancer. So hepatitis B and HPV. It's cheating to say these are anti-cancer vaccines because most people think of them, you know, as preventing the virus infection, but they are really preventive anti-cancer vaccines. So these work just as normal vaccines do by inducing an immune response against something foreign. This immune response is essentially of exactly the same quality and type that you would get if you were normally infected with the viruses, but without the risks of having an active virus in you. If you're interested in hearing more about vaccines, you can type into the comment window or let me know or something, and I can give the vaccine talk at some point in the future. There are also therapeutic vaccines for cancer that are in development or in the clinic currently. For metastatic prostate cancer, so a really, really bad, I mean, prostate cancer alone is pretty bad, and if it's metastatic, it's really bad. The therapeutic option that we have here is sipuleucel. Doesn't work super great. I think the next slide explains this. Yeah. So I'll get back to it a little bit. There's also a melanoma therapeutic vaccine that's I believe in trials currently. And the idea would be you have your tumor already, you have some immune response happening already. So let's boost the immune response, particularly against neoantigens. So these are these new proteins that are really expressed on the tumor and not elsewhere. There are significant challenges with this approach because you have, again, the tumor has all the normal immunosuppressive mechanisms that we've talked about before. So there are additional hurdles. You can make a cancer vaccine in a number of different ways. You could just stick in the neoantigens as recombinant proteins. This has tended to work really poorly because the proteins are cleared pretty quickly for reasons that are not entirely clear to me. But this is the traditional vaccine approach. The sipalucil approach is what's shown here. So you take blood from a patient, you do leukapheresis, so you pull out the immune cells, and then you culture those antigen-presenting cells that I mentioned earlier. Those are the things that activate T cells. So you grow those in a dish. You essentially just pulse them with your tumor antigens, so they're already presenting parts of the tumor, and then you put them back into a patient in the hopes that when they're back in the patient, they'll activate the patient's normal T cells, and the T cells will expand and kill the tumors. So sipalucil hasn't worked all that well, but as I mentioned, it's in a really challenging indication. I mean, prostate is not an easy cancer to go after, and the immunotherapies have had remarkably unimpressive results there. There are also really fancy engineering methods that people are working to make better cancer vaccines. A lot of these, let's see. How does leukapheresis separate out specific cell types? It doesn't. It pulls out immune cells, leukocytes. The thing is, your T cells and a lot of other cells, neutrophils, things like that, don't survive very long in a dish. So you can let the cells that you don't want die, and you can also add in cytokines, proteins that are essentially growth factors that promote the cells that you do want. But I believe this approach, the dendritic cells are cultured long enough in the dish that everything else would be dead anyway. You are welcome. Okay, yeah, so you can also engineer really fancy biomaterials in order to deliver vaccines. As I mentioned, the essentially naked protein approach hasn't worked super well. So people are trying to encapsulate these things in nanoparticles. So this is an academic group. I actually haven't gone back and followed up with whether this was moved to the clinic or anything, although it's been several years now, so maybe it has. So this approach, you have a nanoparticle. It's essentially an inert nanoparticle that the body doesn't really recognize. It's pH sensitive, and the red stuff contained on the inside is the tumor antigens, the things that you're trying to vaccinate against. So under physiological pHs, it stays as a perfectly healthy nanoparticle just floating around your body. If you have those dendritic cells that are present in your body, eat these nanoparticles. Once the particles are internalized, the dendritic cell tries to digest them by acidifying the compartments they're in. The nanoparticles pop open, and the tumor antigens are released from the nanoparticles. The dendritic cell presents the tumor antigens just like it normally would, and then hopefully you activate your T cells. And this is an actual picture of a T cell, by the way. It's not the pretty little circles. This is also an actual picture of a dendritic cell. Immune cells are quite ugly, but don't tell any other immunologist that I said that. Yeah, so the idea would be you enhance tumor-specific T cells that way. So what do you guys think? What approaches do you think are most promising? This is obviously not a, right or wrong question.
Can I answer your question with a question?
Yeah.
In terms of CAR cells, I mean, it seems as though the sort of tailored approach is great, except that price tag. So how long do you think it'll take for people to knock that price tag down?
So the price tag for that approach, I don't think can be knocked down. There are a number of companies now that are trying to make essentially off-the-shelf products that you could take you could take immune cells out of any healthy person, engineer them as needed, and then put them back into any any person that you want, and have them be accepted and do their job. It's challenging to do that with T cells. It's not impossible. People are trying it. But there are additional cells like NK cells that don't have exactly the same recognition mechanisms and might be better suited to that approach. If you could do that, if you could just take healthy cells out of any patient, engineer them, expand them, and then put them into whoever, then this approach will be dirt cheap, because you wouldn't need all of that technology present in the actual hospital to do it.
Right, because that's my sort of thought on that question is like, well, that seems very promising if you can make it affordable.
Right. Yeah, there are also approaches that people are taking to make the CARs in the patient. So delivering, all of the genetic material to alter and generate CAR T cells within the patient. You'd need some sort of nanoparticle or some delivery mechanism to do that. And those are, to my knowledge, not in the clinic yet, but probably will be soon. Those will also be dirt cheap approaches because you just make a nanoparticle and stick it into a person. I mean, that's a really easy thing to do.
Which countries are on the forefront of studying the CAR technology?
US is really good at it. The technology was developed here. There was a lab, Carl June at UPenn, I want to say, or Pitt. Don't quote me on that. So the technology was initially developed here. There are a lot of European labs that are also working on the approach and doing quite well.
Okay.
But yeah, I mean, the more minds we have working on these things, I think the better. So with that, I'd like to thank you and ask if there's any questions. And I do want to reference XKCD one more time. Please, 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 if you're reading Facebook or Reddit or something and you see something that says, some drug or vitamin kills cancer cells in a petri dish, act very skeptically, because you can kill petri dish cells with a handgun. It's not a viable approach for curing cancer in a patient. Just keep that in mind. So what questions do you guys have?
Question about inflammation.
Yes.
What can we do to keep inflammation down in our bodies? Is there anything we can do?
Yes. I'm going to tell you the secret of the only things that we actually know to, what's the claim people put on cereal boxes? Boost immunity or something, which generally also means properly regulate immunity. Number one, don't stress. We know that cortisol causes a lot of immune dysregulation. Not a good thing. Number two, sleep enough for similar reasons. And number three, eat a balanced diet full of, colorful vegetables and fruits and stuff. And it's very clear that malnutrition causes immune dysregulation. It's not entirely clear what parts of proper nutrition properly maintain immune homeostasis. But those are the three things that are actually scientifically proven to properly regulate your immunity.
Thank you.
Yeah, sorry if it wasn't a more emotionally satisfying response.
No, very good. I have one more question, can I?
Sure.
Regarding the someone who is HR2 positive breast cancer.
Yep.
Any info on TDM1 was a trial that was out, and they used it on stage 4 cancers and got very good results. So they've been using it on people like me. And I was wondering if you had any info on that particular trial.
So off the top of my head, I don't know what that trial is, but I'm looking it up, and I think my understanding of this trial is that it's trastuzumab. Let's see. No.
Yes.
Trastuzumab is an antibody that has had some success in the clinic. Currently, it's a tumor targeting antibody. It works by a couple different ways, one of which is it binds to the tumor cells and it recruits these natural killer cells that I mentioned earlier and tells them to kill that cell. Yeah, I've actually
Thank
used that.
you.
I've used that antibody in experiments, just like in vitro
Mhm.
in a dish experiment before, and
Mhm.
it's pretty good at killing cells in a dish.
Okay, thank you.
Sure. Any other? All right. Well, I will stay on the line for a little bit. I'm going to stop the recording now.